Hello, everyone, and welcome to Back Ashore, Schmidt Ocean Institute's live series where we gather some of our past scientists and experts from around the world to discuss topics related to oceanographic research aboard RV Falkor and Falkor 2. Today, we are going to wrap up the year discussing the process of discovering, describing, and naming new species. My name is Allison, and I'm the Senior Manager of SOI's Research Portfolio. I'm coming to you live today from the public library because my internet was not working, so I apologize in advance if there's a little bit of background noise today when I speak. Today we are joined by Dr. Charlotte Seid, Manager of the benthic invertebrate collection at Scripps Institution of Oceanography. Through her role, she supports the scientific process, requirements, and ship-to-museum journey involved in species description. She previously sailed on board Falkor in 2018 off the coast of California in the USA. The Scripps benthic invertebrate collection houses specimens from 7 RV Falkor and Falkor 2 expeditions, from which 10 new species have been described to date. And I've heard she's got many more in the pipeline, so I'm excited to hear from Charlotte. We're also joined by Dr. Jeremy Horowitz. He is currently the Peter Buck Postdoctoral Fellow at the Smithsonian Institution's National Museum of Natural History in Washington, D.C. He studies black corals that occur from just below the sea surface down to the deep sea using an integrated genomic and morphological approach combined with technologies such as 3D imaging to examine the taxonomy of the black corals. Jeremy sailed on Falkor in 2020 around Australia, and he described 4 new species of black corals from those expeditions in Australia. Thanks for joining us, Jeremy. And finally, we have Professor Alex Rogers, who is joining us from Tenerife, and he is the science director of a brand new global program called the Ocean Census. Ocean Census is a global program on a mission to accelerate the discovery and protection of ocean life. I'm very excited to hear more about this program from Alex today. He brings expertise in marine biology, molecular ecology, and the deep sea and extreme environments. I already said he is joining us from Tenerife, where he is in the final stages of one of the Ocean Census's missions to discover some new species, and he'll be talking about that in a little bit more detail soon. During its 10-year history of enabling oceanographic research to take place aboard its vessels, scientists on Research Falkor have described over 30 new species, with many more to be published, which we'll hear about today. As we learn today and hear from Jeremy, Charlotte, and Alex, seeing new to science species on an ROV, obtaining a sample or specimen, bringing it home to the lab, describing its morphology and what makes it unique from other species that are already known to exist, and then formally naming it and describing it in a scientific publication takes a very long time. In fact, the Ocean Census website stated that the average length of time to describe a new species from when it's first collected is around 13 and a half years. So we're going to talk about that process more and learn how our scientists are working to speed up that process as well. I'm going to have each of our scientists introduce themselves personally and talk a little bit about their work. And then I have a few questions prepared, but please feel free to enter your questions in the chat. Charlotte, would you like to introduce yourself and your work first?
Sure. Thank you, Allison. Hello, everyone. I manage the benthic invertebrate collection at Scripps Institution of Oceanography, which is an archive of around 71,000 preserved samples. These are mostly jars of preserved specimens, as well as DNA, ethanol tissue, images, and digital data. So I serve as a resource for researchers at Scripps, but also around the world. I work with Professor Greg Rouse, our faculty curator. I'm often lucky enough to accompany researchers on expeditions, but my primary role is to make specimens available to the global scientific community.
Excellent, Charlotte. And I love the props of jars you have in your background to give us an idea of your work. Jeremy, would you like to introduce yourself and your work to our audience?
Sure. My name is Jeremy. I'm a postdoctoral fellow at the National Museum of Natural History, and I'm a black coral taxonomist, which are these guys. black corals are found all over the world and at all depths. And what I'm doing is integrating traditional morphological data, like are they branched, their colors, and combining that with emerging molecular data, things like phenome skimming and target enrichment of conserved elements. And I use multiple lines of evidence to taxonomically review and revise the order. And a lot of the samples coming from Falker are now being used to work on revisions of all 7 black coral families.
Excellent. Thank you, Jeremy. Alex, would you like to introduce yourself and some of your work?
Sure. Thank you, Alison. My name is Alex Rogers. I'm Science Director of the new Ocean Census program. This is a program funded by the Nippon Foundation and implemented by the Necton Foundation in the UK. Our main mission is to accelerate the discovery and description of marine species globally, and to really communicate the wonder of the ocean and ocean life to the public. And we're out here in Tenerife at the moment, entering the closing stages of our first flagship mission.
Thanks, Alex. I'm excited to hear more about how that mission went. Let's get started with a few questions. I'm going to start with Jeremy. You were on Falkor just a few years ago in 2020, sailing in a very understudied region of the world off the coast of Australia. Could you describe what your process was for seeking out new species, specifically black corals, while you were on board? And were you looking for new species of black corals, or did you happen upon them while you were doing other research?
Sure. Yeah, like you said, they're an unknown group, and we were surveying and searching in an unknown part of the world. So I guess what was cool is that we didn't really know what to expect, and we had very little material to compare what we collected to. A lot of these things maybe have been collected once, more likely never collected before. And so I guess the process was to maximize the usefulness of what we did collect. And so on the ship, the process, I guess, would be to take really good in situ, in water pictures, and collect important parts of the colony, that are informative and useful in identifying species, genera, and families. Image the corals on the ship after they come aboard, and carefully subsample the tissues for downstream DNA analyses. And we take images of really small sections. In this example, in this picture, we would take a 1 centimeter fragment, remove the tissue, and use a scanning electron microscope, which allows us to look at the spines, the skeletal spines on the colonies, which are very informative at the species level. And so we use the images, the fragments, the data, and we use all this to make informed taxonomic decisions. I guess explicitly, we were looking for new species, or at least I was, on these expeditions, because so few people work on this group, and most of the species are deeper than you can't scuba dive and collect most of these specimens. So we're really, for the first time, seeing these species in their natural environment, and they're really spread out over the ocean. So it'd be really hard to collect it by dropping a trawl and dragging it across the seafloor. You'd miss a lot of them. And so I guess we're able to fill these really large and fundamental knowledge gaps for this group. I enjoy it. I like it because they're really mysterious. And I guess we now have the technology to fill these large fundamental gaps, and we get a lot of bang for our buck, because, with very little effort, we can finally answer these long-standing questions about how they're related and how many species are in different regions, and what makes species different.
It's great, Jeremy. I'd love to talk more about the technology advancements and some of the excitement that you feel when you came upon those species. Did you know they were new species when you saw them on the seafloor, or did it take some time once you were back in the lab to kind of verify your feelings that these were new species?
It's really hard to identify something as new unless it looks really, really different. But often the boundaries between species are things that you really can't see with your naked eye. Often branching pattern is misleading because a certain branching pattern will independently evolve multiple times in different parts of the world, different times in history. And so often, I guess you're just excited to collect something that looks different. And if you collect enough different looking things, you capture the diversity in a region. And then later on, when you have images and you have, sequence data, at that point, you could start to decide which are new species and describe them.
Very exciting. Charlotte, we mentioned that you have sailed on board research vessel Falcor before, but as a collections manager, you often receive specimens that you yourself did not collect. Could you describe what your process is and what happens when species or specimens come to you and you think they may be new to science? What's your process in the lab?
Yes, it's always quite a process. First, physically getting the specimens from someone's ship or someone else's lab into my hands can be a major victory. The specimens don't ship themselves, so very carefully packaging them and complying with all applicable regulations is a big part of my job. But I also treat every specimen as if it were potentially new in terms of data quality and looking out for its physical integrity. So the slides here have a few wonderful images of representative new species, but we didn't always know they were new at the time. So ensuring that the collection has the data, latitude, longitude, depth, date, the person who collected it, the ship, the vehicle, etc., all the information that Jeremy and folks might have painstakingly recorded needs to come to the collection in digital form, so I can put that into our database and therefore into online aggregators and to places where anyone in the world can publicly search and then find those specimens for further work. I'm also concerned with the physical preservation of the specimen. If it came to us on dry ice and there's a really important tissue sample for, say, genomic work, we want to maintain that cold chain. If it was preserved in ethanol, we need to make sure that that stays at the appropriate level and concentration. So every specimen is precious. We need to give them all the star treatment. And even if they're not new, having high quality reference material from known species is just as important in putting this whole biogeographic and taxonomic picture together.
Thanks, Charlotte. That's a great point about preserving known species as well, so you have something to compare them to. I also love your statement about treating them like the stars they are. That's really exciting when you get something in the mail, I guess you could say. Alex, you mentioned you are out on an expedition right now with the goal of describing and finding new species. Could you give us a little sneak peek about what you found on your mission and what the goals of the Ocean Census program are while you're out there in Tenerife?
Yeah, certainly. I mean, Tenerife is quite a challenge for us. The Micronesian archipelagos have been studied for about 150 years. So you wouldn't expect that many new species to be still there to be discovered. What we've been doing over the last 3 weeks is using a team of divers from the Finnish Academy of Scientific Diving and the Pisces 6 submersible, which I think was built back in 1976, but was completely rebuilt in 2015, to sample various habitats down to about 250 meters around the islands. We have discovered dozens of new species. So the one you just saw is an Alphaeid shrimp. There's more than 300 species of Alphaeus found globally. They're very important animals in ecological terms and produce much of biological noise that we hear when we're snorkeling around here. This is a Nemertean worm, my specialist group. This is a Cerebratulus species, probably new to science, but we're sending an example off for DNA barcoding. We've got other examples for morphological taxonomy. This is a Fellaini species type of mollusk. And just to say, the thing that unites many of these taxa is that they're very small. Some of them are only a few millimeters long. And what we're finding is that it's almost a situation of the closer you look at these marine ecosystems, then the more that we're finding in terms of species. This is Balanoglossus, probably a new species. We need to confirm that because it does resemble another species called Balanoglossus minutus, but we believe it has some characters which differentiate it from that species. The Ocean Census approach is to use a variety of modern technologies to try and accelerate the whole process of taxonomy. Those include using digitization to really digitize all the information associated with the species we're finding to really enable cyber taxonomy down the line. And we're actually almost differentiating the discovery process and the species description process. Our intention is to really publish what we call digital life forms, where we have good images and other information, and an expert taxonomist says, I believe this is new, and genetic information as well. And then further down the line, the species is described. But what that means is that ecologists and other taxonomists can actually look at the data we're producing and say, yes, this looks like the same thing that I've just found in a cod stomach or whatever. So we're trying to really push the envelope in terms of how fast this work is done. And I'm very happy to report that some of the animals like this is a new species of Giberula, a member of the Cytiskidae family. And you can see again, it's a very small but really beautiful animal. We've been able to actually go from animal being collected to draft publication, so a full electronic paper in the space of 48 hours in some cases, which I think might be a record. As you mentioned at the start of this, the average length of time for a species to be described is about 13.5 years. But it shows if you have experts on hand, you can really turn over this species discovery and description very rapidly indeed.
Thanks, Alex. Those images were beautiful, and you prompted my next question for Jeremy really well. He actually published his new species of black coral about 2 years after he found them in Australia. And I was about to say that that was a relatively quick turnaround for naming and publishing, but you have proven me wrong with the 48-hour turnaround. But nevertheless, Jeremy, you did come out with a paper about 2 years after you sailed, and so I was wondering if you could talk about your process in validating these new species once you were off the ship and publishing on, let's say, a quicker than average timeline, even if you weren't able to set a new world speed record for publishing. I think in the scientific community, it was still a pretty quick turnaround. If you could talk about your process from coming off the ship to publication.
Sure. Yeah, as Alex said, when you have the experts and the technology all under the same roof, you can do it all really quickly. And so when I published that paper, I was in Australia associated with the Museum of Tropical Queensland, and now I'm here at the Smithsonian Institution. And these are both places with really large collections of black corals. Specifically here at the Smithsonian, it's the largest black coral collection in the world. So most species, we have representatives here. So I have the whole order, all of the black coral species within an arm's reach. And that also means I can shave a lot of time. Hannah talked about the red tape in sending samples. You have to package samples. Well, first you have to contact the curator at a different museum if they have a sample that you need to compare to what you have to determine if it's new. And, you need them to find the time, the curators to find the time to image it or subsample it or send it. And then you have to, it takes time and money. When you have everything all here, you're saving a lot of time, a lot of money. So we recently were in Puerto Rico on Falker 2, and we brought the specimens back. And then weeks later, we have evidence to describe new species. And so I think when you have the experts and the resources all together, it's not as hard of a process. It's pretty straightforward and streamlined.
That's a great explanation. I did not know that about the Smithsonian. I'll have to venture down and see all of the black corals that they have in their collection. Charlotte, you're kind of at that nexus that Alex and Jeremy both talked about, where some of the collections and the science and the naming kind of all live under one roof. Could you talk a little bit about kind of how you and your role bring all of those different components under one roof?
Sure. I'm also happy to touch on why it doesn't always go as smoothly.
Exactly. They made it sound really easy.
So first, I would say the process of describing and then critically publishing that new species is an exercise in due diligence. Often we're very lucky to work in understudied areas of the world or the deep sea where very few humans have gone there before, and it's fairly obvious that something is going to be new and you know what the relevant comparative material is. If you're working in a more crowded field or species that might have been described 100 years ago and you're not sure how yours fits into the puzzle, that can involve a lot more detective work, even if you're lucky enough to have your own materials on hand. So in addition to the careful genetics and morphology that my colleagues here alluded to, there's a lot of very careful reading of the literature. Often you have to go back hundreds of years and look at papers from the 1800s, compare their animal to what you have on hand, to make sure you're not overwriting someone else's work, that you're not describing the same species twice. The burden of proof is on you if you think you have something new. And it's also important to put it into a broader framework. We're talking about species as individual units of biodiversity, but ideally we'd like them to fit into a higher taxonomic framework. Jeremy mentioned genera, families, orders, generally a higher taxonomy that we think should reflect evolutionary relationships. We think there should be a consistent way of grouping like with like and relatives with relatives. But historically, this wasn't always done in a consistent way. Genetics wasn't a thing in the 1800s. So a lot of this higher framework is done in an inconsistent way. So when you want to add your one species to the family tree, you unravel a mess. And we like to try to sort that out before proceeding, so you know what genus your species goes into and maybe shed some light on the rest of its evolutionary history. So that takes often comparative work. Maybe you need to request a specific example from a neighboring species. Often it needs to be from a specific part of the world. Which one is the real one? Biogeography is an important consideration. So if you think you have a new species, but there are a lot of lookalikes in that area, that might be a cause for extra due diligence. Whereas if you think you have something that's found in another ocean basin, you might want to be a little extra careful in claiming that it's a range extension of definitely the same species. So integrating genetics, morphology, biogeography, and literature can be a really academic exercise in critical thinking, but it's nonetheless important to do it right. That's my soapbox. But once the specimens do go through that process, one of my favorite parts, of course, is having them deposited in a museum. Strictly speaking, the most important requirement is designating a holotype, one example that defines that species, becomes the gold standard, the reference for anyone else trying to figure out what defines that scientific name. So that's a pickled thing in a jar that goes to someone's collection, or in rare cases, it can be a very high quality image if the physical specimen doesn't exist. So that's a really important privilege and responsibility of my work, to catalog those specimens and then make them available for future detective work requests, but also look out for their long-term archival. I can't just give them away. I need to make sure they come back if they're loaned. So really maintaining that library and fitting everything into a coherent framework is why this is a more difficult exercise than you might imagine.
Thanks, Charlotte. Some of the images that flashed up while you were speaking had names of the new specimens, and I know that you have played a role in naming some of the specimens that are described at the Scripps Institution of Oceanography collection. Could you describe your thought process and what it's like to actually name a new species? And do you have a favorite new species that you have named?
Sure. As an example, I love to use my friend Munidopsis gurgasi. This is a squat lobster, a crustacean described from several Falkor expeditions, but the one near and dear to me was in 2018 off Southern California. So our collaborator, Pete Gurgis from Harvard University, was the chief scientist, and many labs were represented. We were studying the animals and microbes and habitats off some of the most densely populated areas, Los Angeles, the hashtag backyard deep, and what lives offshore this major metropolitan area. So we found these adorable crabs. They were bright orange in real life with a little rocket ship marking. They were really distinctive. I couldn't immediately match them to anything known, but knowing they might be potential holotypes, I took lots of pictures and very carefully logged them and all their data. Luckily, a few years later, we had a collaborator, coincidentally, from Harvard University studying this group from around the world. So she and I and our respective curators went on this big scavenger hunt, combining molecular and morphological data from all these squat lobsters. It turned out that the creatures I had collected on Pete's expedition matched something from the Harvard collection, as well as a specimen from our Costa Rica 2019 expedition with Schmidt. And we ended up naming them in honor of Pete Gurgis, since he was so enthusiastic about collecting them. And they happen to fit with the microbes and the area of the world. So I would say this is one of my favorites because I was part of every step of the scavenger hunt.
That's awesome. And I know Pete Gurgis very well, and I'm sure he was honored to have a squat lobster named after him, and I hope he's watching today. While we're talking about naming new species, Jeremy, would you like to share some about the black corals that you have named and what your process was for naming them, or some of the reasons behind the names that you gave, and if you have a favorite one?
Sure. If okay, I'll talk about two. The first one, you had a picture of it earlier on like a nautilus shell. While we were out in the Coral Sea, we collected this really cool, actually it looks somewhat similar to this one. There it is there. And we went to collect it, and it was under sand, this nautilus shell. So we just thought we were collecting a rock that it was attached to. And luckily, we had a really good ROV pilot who carefully picked it up without really damaging the nautilus shell. And it ended up being a new species. So this is a relic species on a relic species, which is really cool. And I thought it was so cool that I named it after my grandfather, Baikovsky. So I called it Baikovskii. So that's probably one of my favorite moments on Falkor. And then the other, I named it after Falkor to thank them for allowing me on the ship. And I also worked when off the ship. I'm not sure how happy the chief scientists were, but I always like to be involved in expeditions and, when possible, ask for material to be collected. And so this was collected, I think, at about 100 meters. And I named this one after Falker because it had the coolest skeletal spines, which are these features on, so each one of these are branches, and on the branches of these skeletal spines. And these are the coolest skeletal spines on any black coral species, I think. And so we called it Antipathes falcorei. So those are my two favorite.
Awesome. And I believe the falcory was also named one of the World Register of Marine Species top 23 species of 2023. So that was exciting for that really unique structure you just talked about. Alex, it sounds like you are about to come home with a lot of species to be named on your plate. We actually have a question from the audience about how to ensure respectful collaboration when naming new species, particularly if you want to reference maybe another indigenous nation or the local community from where the species was collected. And so since I think you will be naming some new species soon, I was wondering if you could speak to that question.
Absolutely. We've thought long and hard about this when developing the Ocean Census project. Our principle is co-planning and co-production of our expeditions with coastal states where we're working within the exclusive economic zone of nations. Here, of course, we're working in the Canary Islands, and we have local Canarian scientists working on the expedition, as well as scientists from the Cape Verde Islands and a sort of broader Northeast Atlantic region, and from mainland Spain. The way we work is actually sort of going to turn over the traditional model of how these expeditions have worked in the past, in that we aim to deposit our material in a regional or national museum where we are working. So all of the material from this expedition is not coming home to Oxfordshire. It's going to be deposited in the Natural History Museum here in Tenerife. Likewise, we have an expedition coming up in New Zealand. The material there will be deposited in NIWA, the National Institute for Water and Atmospheric Research, and the Te Papa Museum in Wellington. We will have some of the indigenous Maori tribe people on the vessel. And, you know, we sincerely hope that they will actually be naming some of these species as they're coming on board. And we very much want to kind of expand this process as Ocean Census continues in the future, where we're working with indigenous people and depositing material basically in those states which host our research to really create a national cultural legacy in terms of taxonomy, and to move away from this kind of colonial approach to marine taxonomic work that obviously was a feature of the great expeditions of the past, things like Challenger and some of those other sort of global expeditions.
Thank you, Alex, for that. And I appreciate the mindfulness of making sure the specimens stay there in the country to expand the knowledge that local scientists can have of their own region. So that is very important. Charlotte or Jeremy, do you have anything to add to this audience question about respectful collaboration when naming and describing species?
I would say we take the same approach. Often the permits or collaboration agreements require that type specimens of new species be deposited in the home country. So we're absolutely happy to do that. It's best for the community, even if those valuable resources are distributed, should anything happen to one collection, or should there be requests from one country versus another. So often that's another round of shipping, another round of coordination, but we're absolutely happy to do it. And then keeping track of those requirements, linking them in our database. So if someone does request that specimen, they make sure they credit the Indigenous community or the home museum appropriately.
That's great to hear. Like you said, it's a lot more shipping and sometimes more work, but I think it's really important to be respectful of those collaborations. Jeremy, did you have something to add?
I think just echoing what they said, naming things based on, if you want to name it after the location it was collected from, it'd be really nice if you find the local name based on, Aboriginals in Australia, Indigenous people, to make sure they're happy with, respect them and make sure they're happy with the name. And also depositing specimens in local museums is a direction that I think everyone is now moving towards, to allow local scientists to have access to the material and to, remove that barrier. Otherwise, people don't have the money to travel to the few large museums in the world that house most specimens. So trying to level the playing field is a really good thing.
Definitely. And Charlotte, I wanted to go back to something that you said in your response that's related to another audience question just about shipping and transporting some of the specimens within country or to museum collections. We had an audience question about whether there are specific import or export laws or hoops to jump through when shipping specimens from the ship. And I see Alex laughing because I think he's about to do a lot of this with his mission just ending, but maybe we'll start with Charlotte on this one.
Yes, a well-anticipated question. I would say that the very first concern in my mind is often CITES, the Convention on Endangered Species, which is certainly near and dear to Jeremy as well. So shipping endangered species internationally rightfully should be viewed with some scrutiny to ensure that unscrupulous things aren't happening. Registered institutions such as museums and universities will usually have a certificate of exchange. So it's additional paperwork, and someone like me has to file that online, link the specimens to their collection permits, to the exact shipment, all that paperwork, and then ultimately to this endangered species permit if we're working with endangered species. So it's a burden, but it's ultimately important. For non-endangered species, there are also hoops to jump through. They're often very country specific. So having local knowledge, say at an international museum, can help make that process smoother. Within the United States, the U.S. Fish and Wildlife Service has a process for declaring wildlife. Even if it's not alive, even if it's sterile and preserved, it still needs to go through this paperwork process. So again, I'm happy to do it, but it's just one more step. It takes time and it requires solid data and cooperation all around.
Thanks, Charlotte. Alex, do you have anything to add to that?
I think so. I think it's important that we respect various international treaties that have been established around biodiversity. I mean, nobody likes paperwork, but things like the Nagoya Protocol and the upcoming Biodiversity Beyond National Jurisdiction treaty really require us to declare what's been recovered during an expedition, and then to really have traceability of those samples as they're moving, through countries or from institution to institution and so on. I think a very important point to make here is that through working with regional institutions, you can actually avoid some of that paperwork. But also, I think an important point to make is that the digitization of material now, the ability to create really high resolution, three-dimensional imagery of taxa. I mean, with Jeremy's really wonderful electron micrographs of these black coral skeletons, I mean, you can imagine, getting a lovely three-dimensional image of that organism, which enables a taxonomist and the other side of the world to examine that material without it having to be sent across borders with all the paperwork and everything for it as well. So I think digital technologies and these approaches like micro CT and so on are really going to enable us to start adopting cyber taxonomy as a standard for many organisms, and help us to really, enable scientists anywhere in the world to study material held in one locality without going through that pain and cost of transporting material.
Thanks for bringing that advancement up, Alex. I happen to love doing paperwork, but I'm all for new technologies that make it easier. I'm just kidding. Nobody likes paperwork. But Jeremy, Alex mentioned some of the technologies that you used. Would you like to add anything?
Yeah. I guess to continue on this talk of the three-dimensional models, it's actually something that the Smithsonian Institution is really pushing for, because a lot of our holotypes, a holotype is the specimen from which a species was described, so it literally represents the species. A lot of them are here, and by making a 3D model of the specimen, you can send that in an email to someone who's trying to compare their specimen to a holotype to determine whether it's new or not. And so, yeah, immediately you can get this amazing high resolution digital representation of the species and make use of it. So it will really speed up going about revising taxonomy. And it also makes it more accessible to people who don't have the resources to visit a museum.
Thanks, Jeremy. We have another audience question related to shipping. Apparently, this challenge of getting specimens around the world is a very intriguing process to our audience, which is fine. But some people asked if there are specific companies that you use for shipping specimens, and is it scary to ship such valuable scientific specimens or commodities around the world because of risk or damage, or worried about it getting lost in the mail, so to speak. So I'll open this up to anyone if anyone wants to take this audience question.
Well, thank you for appreciating the less romantic aspects of the job. So usually the major world couriers are the ones used, but that can be country specific. Some countries prefer DHL versus FedEx or UPS or what have you, National Post. So rather than any specific company, it's what makes sense in context. But there are international rules in terms of how you can package things. There's a special regulation, IATA Special Provision A180 regarding museum specimens and how much fluid you can have per jar and per container. So 30 milliliters of ethanol, formaldehyde, or mixtures thereof. So there's a lot of arcane details. And once you get used to it, it's pretty straightforward. You need to heat seal them to prevent leakage for flammability purposes, et cetera, et cetera. So yes, fortunately, there are some good international rules and you need to put the appropriate verbiage, but nonetheless, I do always have a little bit of anxiety with every baby going out into the mail. Sometimes you get that one customs inspector who has more questions than the last person. So yes, it's not to be taken for granted. Thank you for appreciating that.
So everyone, when you're thinking of shipping out holiday packages and standing in line at the post office, just imagine that you could be shipping a squid or a squat lobster or something far away. So it definitely brings a new appreciation to the process. Alex or Jeremy, do you want to add anything to the fun shipping conversation before we move on to a few other questions?
I think it's always quite challenging and always a little scary. We have a favoured shipping agent we use, but just to give an insight into how complex this whole thing has become UN regulations on transport and materials now specify the exact boxes you've got to use, even down to the tape you use to seal up the boxes. So we actually have to ship that material out to a locality to ship material back in the right boxes with the right tape and the right hazard labeling and so on. So it really is, as Charlotte said, a very complex business.
All right, well, thank you for that, and thanks to the audience for that question. We've talked about this quite a bit, about kind of speeding up the process of how we name species using new technology, using imagery that we can just email. But I wanted to delve into that a little bit more. Alex, the Ocean Census website also says that around 2,000 species are discovered each year, and that rate hasn't changed since the 1800s. So could you explain a little bit about why Ocean Census was formed and what you are aiming to do to speed up this process of new discovery? I know we've touched on pieces of that in various questions in our conversation so far, but maybe we can package it up nicely for a good conversation about how we speed up this process.
Sure. We actually thought for about 2 years as to how to do this when carrying out feasibility study for the Nippon Foundation on how to accelerate species discovery. I think the bottom line is that for many years we've been actually kind of struggling along with 19th century science approaches to species delimitation and species description and so on. And now, because we're facing a biodiversity crisis in the ocean as well as on land, it's become extremely urgent to understand more about where life and what life is distributed throughout the ocean. I think we've got to bear in mind that the ocean is this treasure trove of 4 billion years of evolution on Earth. And every species is precious in its own way as a unique product of that evolutionary process. So for us, this is a really urgent mission. I've already mentioned digitization as being one of the core aspects of our strategy, but integrated taxonomy is very much a priority for us. So this combination of the use of different technology approaches in species discovery and species description. So DNA barcoding or genome skimming, use of digital imaging, three-dimensional imagery, even down to the use of new technologies for sampling environments and organisms that using conventional sampling methods are really difficult. And I'll just give you a trivial example of that. Anybody who uses push cores in the ocean from submersibles or ROVs will know that sediments which are quite unconsolidated are a real nuisance to try and sample. So recently we developed with RevOcean a new coring tool which actually retains unconsolidated sediments in the core tube, whereas normally they'd just drop straight out. And that means we can study the organisms living in a habitat that previously has been very difficult to sample. So it's really an all-points approach in terms of technology, but another core part of what we're trying to do is increase capacity in marine taxonomy globally. As I said, we have this co-production of science approach, and what we hope to do is go to countries around the world and actually work hand in hand with early career researchers from different countries, develop their skills in taxonomy and enable them to describe the species living in their waters and contributing to their own museums, contributing to the understanding of the marine ecosystems that are a priority to their countries. So it's a combination of approaches: digitization, new technologies, integrated taxonomy, and increasing capacity. It is the way that we're trying to do this.
Well, that sounds amazing, Alex. Some very lofty goals, but I'm excited to see where the Ocean Census program takes all of this new species naming and their initiatives in the next few years. I think it's really exciting. Jeremy, you had mentioned some initiatives that the Smithsonian was undertaking kind of related to this as well. Could you talk about those a little bit?
Sure. All of the holotype specimens are being imaged, and they're going to be made publicly available. So all you would really need to do is go onto their website and just download the model. And we're starting with the most important samples, these holotypes. And once those are done, I mean, the goal is to digitize the entire collection. At the same time, we're also making available molecular data and really robust, full genome, these really powerful, this really strong molecular data also to be publicly available. So you can go to their website and click on a specimen name. And what will pop up is a link to GenBank, where all the molecular data is deposited. And if a 3D model is available, you can just click on the link below and look at the actual specimen. There's also in situ images that are also in this same page. So you've got all the information you need all in one place. And so I think taking advantage of the technological advances really allows us to overcome lots of these barriers and publish much quicker, and have the data much more easily. So it's very useful.
Thanks, Jeremy. Yeah, it sounds like a lot more is able to be made publicly available to a lot more information and data, not just the specimen that you might want to hold. So that's really exciting. Charlotte, would you like to share anything new at the benthic invertebrate collection related to some of these technological advancements?
I'd say we're all aligned in our priorities and mission. One ongoing project with this collection and around 19 others around the US is digitizing our historic specimens. This is an initiative funded by the National Science Foundation called Dig In, Digitization of Invertebrate Collections. So we're not even doing micro CT and all of that, simply getting the information on the labels from paper to digital, catching up on hundreds of years of backlog in a concerted fashion using the same data fields, and sharing our problems and triumphs among collections has been an ongoing project. So it's really great to see the invertebrate zoology community coming together and making that happen. I also wanted to hop on Jeremy's last statement about publishing. Often that can be the most exciting, triumphant part, but it can be a sticking point as well. Once you've compiled all this beautiful data and finalized your paper, it goes off to a journal, but getting it actually published and formatted can be another journey. Having your peer reviewers, fellow scientists, review the quality of the work you've done. Do they believe it's a new species? Do they think further science is needed? That can be a really lengthy process, especially since peer reviewers aren't paid to do this. It's a volunteer service that we do for the community. Many folks don't realize that scientists often pay to publish their own work. It's part of our job, but we need to come up with the money to make these papers available. So I do want to give a shout out to Schmidt Ocean Institute for funding open access publications. Often it's thousands of dollars to get your paper out there, but if you put up the money, generously supported by Schmidt or others, then that paper is free for anyone on the internet to read. But if you don't, then it's on the reader, and that's another barrier to knowledge.
Thanks for that acknowledgement, Charlotte. And yes, we are very happy that we are able to support the open access of Falcor-related research so that anyone in the world can access the scientific outcomes of the research. And that's a very important piece of this new species journey that we've been on over the last almost hour. We do have a few audience questions. So the first one is about DNA sampling, and if DNA analysis is done on a new sample, can that be compared to the DNA record in an existing collection rather than needing the physical specimen to compare? So comparing DNA records against DNA records, is that a better proof, I guess you could say, of new species? I'll let anyone take that question.
Yeah, so what makes it really useful to compare genetically specimens, once you have a phylogenetic tree, once you have, it's like a family tree of how all the species are related, you can simply add a new specimen to that tree and see where in the tree it falls. And it may fall exactly where you expect it to because the branching pattern is very similar, or it may be in a completely different part of the tree, which would be surprising, but it's becoming less and less surprising now that we see this over and over, that these certain morphological features are actually really confusing and independently evolving. So it's really important that you do compare molecularly specimens that you think are new species to nominal or currently described species, and have confidence in that genetic data as well. So yes, it's important, and it is how it's done. You compare your genetic data to other specimens to determine how closely related your specimen is to already described species.
Fully agree with everything Jeremy said. I'd also like to note that there is no universal cutoff or a percentage that tells you this animal is definitely the same as this one. The percentages that are generally accepted can vary. It depends on which gene you use. Some animal groups use different genes, so it's not something an algorithm can do for you. It often requires due diligence, looking at the literature. So absolutely, we love to have those genetics, but your information is only as good as your references. If it's a 100% match to an unidentified worm, well, then maybe you have the same thing, but it doesn't necessarily get you a name. But it's absolutely critical, and we fully support putting sequences online in public databases so this exact kind of comparison can happen.
Thanks.
I think a really important point to make is that getting sequence data now is technically more easy for many groups of organisms and far cheaper than it used to be. So this is giving us access to a much larger part of the genome of organisms than we previously would have been able to access. For some organisms, such as octocorals, for example, gorgonians, because they have a DNA repair gene in the mitochondrial genome, conventional barcoding genes such as cytochrome oxidase 1 don't work very well for that group of organisms. And being able to now do a low coverage genome sequence, a genome skim of those organisms, is really revolutionizing our understanding of their evolution and taxonomy.
Thanks for that, Alex, and for everyone. And thank you to our audience for participating. We have just a few minutes left, and so I would like to wrap up by asking each of our participants if they could share what they're working on now. And then we'll close out with a thank you to everybody. So let's start with Jeremy.
Still working on a lot of material from previous Falker 1 expeditions and our recent Falker 2. One of the exciting findings that this just happened last week, we got sequence data back for 2 specimens that had these question marks on their family ID, which is pretty rare, and they ended up being sister to next to a family that we thought it was within. So it's a very novel branch in the phylogenetic tree. So it suggests molecularly that it's not just a new species or a new genus, but it actually is an entirely new family. And after finding this out, we went back into the collection and we found two other specimens that look just like this one. And morphologically, there are differences between these two specimens and all currently described species. So it's very likely that in the near future, we'll describe an entirely new family based on these four specimens. And it's because we had really good, fresh data from the Falco expedition that's from Puerto Rico.
That's very exciting, Jeremy. Thanks for sharing that breaking news with us. Charlotte, what are you working on?
Likewise, we have hundreds of years of backlog and many years of Falcor expeditions to catch up on and identify. Watch this space for a new genus of deep sea worm from the methane seeps off Costa Rica. This should be published very soon. Also, thanks to fantastic live video from Falcor and specimens collected previously. So another star in our collection.
Excellent. Alex, what are you working on?
Oh, my job over the next few days is writing the expedition report, getting our specimens to the Natural History Museum in Santa Cruz, here in Tenerife, and the rather odious task of packing containers. We've got 2 lab containers donated by Open Cell, packing those and sending them back to the UK, and then preparing for our next expedition, which will be with the National Institute of Water and Atmospheric Research in New Zealand to investigate the Bounty Trough, which is very, very exciting, and I'm expecting a rich haul of new species from that. But I'd just like to add, if there are some taxonomists in the audience, then please take a look at our website and join the Ocean Census network so you can share in this great mission to help describe the ocean's biodiversity.
Thank you, Alex. And with that, I think we'll say happy holidays to all of our guests. Thank you for joining, and thank you to our audience for tuning in, and we'll see you in the new year. Thank you, everyone.